All right, thank you for the introduction. I'm from UCSD. I actually just defended my PhD a couple of weeks ago, so I'm really excited to be thanks guys. So I'm really excited to be here to share with you a little bit of work from one of the chapters of my dissertation. So in this body of work, we were focused on studying the combined effects of multiple enhancers working together to regulate a common target gene. And enhancers are a type of cis-regulatory element that are often focused on in studies of gene regulation. They're known to have very large effects on gene expression with widespread downstream biological consequences. So, getting a better idea of their regulatory behavior is an important task in the field of transcriptional regulation. Now, when multiple enhancers for a gene are active at once, it's typically assumed that they work together additively, meaning that their total effect on their target gene's expression is about equal to the sum of the individual enhancer effects on that gene. However, this isn't always true, and there have been reports of cases where enhancers work together non-additively, meaning that their total effect on the target gene expression deviates from what would be expected under an additive model of activity. Now, it's not clear whether these cases represent the presence of some sort of synergistic-like interaction effect that arises from the co-activation of certain combinations of enhancers on top of an additive model of activity, or if they reflect a different underlying model of enhancer activity altogether. And studies that have looked at the co-regulatory effects of multiple enhancers on the same gene have typically examined specific gene loci, meaning that their findings don't generalize to the entire genome, and have also reached different conclusions regarding the presence of interaction effects as well as the underlying model of activity. So it would appear that the field is limited by inconsistent findings and a limited scope of studies. So here we performed a genome-wide interrogation of interaction effects between enhancer pairs and also evaluated the fit of different models of enhancer activity to try to come up with a generalized conclusion based on the entire genome and to address this knowledge gap in the field of gene regulation. So in order to do this, we reanalyzed data from a single-cell CRISPR screen targeting enhancers published by Gasparini et al. In 2019. You guys, a lot of people have heard mention this data set already. But in this study, the authors designed pairs of guide RNAs to try to repress putative enhancers throughout the K562 genome, with the goal being to try to identify enhancers and map them to the target genes. And importantly, in this study, the authors transfected the cells with a high MOI, or multiplicity of infection, which resulted in every cell receiving a random combination of multiple guide RNAs. And then these cells are profiled with single-cell RNA-seq. And then to evaluate enhancer effects, the authors tested for differential expression of any genes located within a megabase of any enhancer that was targeted in their experiments, basically comparing the expression of these genes in cells that did receive a targeting guide for the enhancer versus cells that did not. Now, in this original study, the authors were focused on the effects of an individual enhancer, but we wanted to leverage this unique high MOI feature of their CRISPR experiment to try to answer our question about how multiple enhancers work together, because all of the cells in this experiment have multiple enhancer perturbations. Now, in a CRISPR experiment, guide RNAs are really important. They are responsible for guiding the gene editing machinery to the intended target site in the genome. However, guide RNA on-target activity and specificity and efficiency can vary based on a lot of different factors, and different guide RNAs are going to behave a little bit differently. But in the experiment I just described, the authors designed pairs of guide RNAs to try to repress each enhancer in their study. But when it came to actually evaluating the data, the authors looked at only whether or not a cell had contained or did not contain a targeting guide RNA for an enhancer, without distinguishing between which guide RNA the cell received. And this approach could be problematic because it assumes that two different guide RNAs function equivalently when this might not necessarily be true. And to investigate this, we calculated efficiency values for all of the guides used in the Gasparini experiments. And indeed, we saw a range of different efficiency values across their guide library. And so based on this finding, we saw a need for evaluating guide RNA efficiency when analyzing data from CRISPR experiments. And to further illustrate this point, we performed a CRISPR experiment trying to repress two different enhancers of the gene NMU that were reported in the original Gasparini study. And so we simultaneously targeted both of these enhancers using the same guide RNAs that were also used in the original experiment by transfecting cells with vectors that contain different combinations of pairs of these guide RNAs. And what we saw was that cells receiving the vector containing the more efficient guide RNA A1 showed significantly greater fold change in NMU expression compared to cells that received the equivalent vector but with the less efficient guide RNA A2 instead. And so what these findings demonstrate is how the joint effect of a pair of enhancer perturbations could appear smaller or larger than in reality if one of the targeting guides had a low efficiency, but we assume expected behavior simply because we saw that the guide was present in the cell. And so to avoid this pitfall, we plan to develop models for evaluating enhancer effects that do account for this important variable. Specifically, we're going to try to do this by calculating a target site perturbation probability for every cell, which is calculated as a function of the efficiency values for every guide targeting a given target site or enhancer that are present in the cell. And what this value represents is the probability that the target site will be successfully repressed with CRISPR-I. Now, in order to evaluate the data, we use a generalized linear model. And as the name suggests, this is a generalized form of an ordinary linear model. And one of the assumptions of these models is that the response variable shares a linear relationship with the predictors. However, this assumption doesn't always hold true. For example, if the response variable is discrete or bounded in some way, as in the case of raw RNA sequencing count data, which is what we're going to be analyzing in this project. And so when you do RNA sequencing, you're essentially just counting the number of reads that overlap a gene body region, which results in discrete and non-negative count data that's been shown to follow a negative binomial distribution. And so GLMs are handy for cases like these where the data doesn't satisfy all of the assumptions of an ordinary linear model. And what a GLM does is it assumes that the response variable comes from some statistical distribution whose mean, mu, is defined by some function of the linear predictor. And this function is referred to as the link function and is indicated here by the term G inverse. And in the case of a GLM for negative binomially distributed data, it's standard to use a log link function, which can be expressed like so, such that the mean of the negative binomial distribution from which we assume the response variable follows is equivalent to the exponentiated form of the linear predictor. Now, this slide shows our model formulation for our interactions model, which looks at pairs of enhancers, A and B, and then determines whether or not they exert an interaction effect on the expression of a putative target gene, which is modeled as the response variable. And so the mean of the negative binomial distribution, which we assume the response variable follows, is defined with the canonical log link function, as shown here, and includes the following terms. We have an intercept term. We have beta A, which represents the effect size of enhancer A on the gene's expression, and XA, which is the perturbation probability of enhancer A in this CRISPR-I experiment. We have those same terms for enhancer B. We have beta AB, which represents the effect size of the interaction between this enhancer pair, and XAB, which is the joint perturbation probability of both of those enhancers. And then we also account for a couple covariates like cell cycle score and percentage mitochondrial DNA, and also include an offset term to account for variable sequencing depth across cells. And then to determine whether or not this enhancer pair exerts an additional interaction effect on the gene's expression, we fit both a null and an additive model to the data and compare the model fits with the likelihood ratio test. And so this null model assumes that there's no additional interaction effect on the gene's expression and essentially removes the interaction term from the model. And so if the alternative model, which does allow for an interaction term, fits the data better than the null model, then this would suggest that there is a significant interaction effect arising from this enhancer pair. Now, having established the formulation of that model, I would also like to discuss the biological meaning of it as it pertains to the question of enhancer dynamics. When we use this canonical log link function for a negative binomial GLM, we're equating the mean of the response variable to the exponentiated form of the linear predictor. And according to the product rule for exponents, the exponentiated form of a sum can also be expressed as the product of the exponentiated forms of the individual terms in that sum. Therefore, it can be said that our model represents a multiplicative model of enhancer activity where the total effect on gene expression is a function of the products of the individual enhancer effects. Now, when looking for candidate pairs of enhancers in the Gasparini data to apply our model to, we imposed a distance threshold of 1 megabase from each enhancer in the pair to the putative target gene. This is because we expect to have the most power for detecting cis-regulatory elements, which are typically located in the region surrounding the gene they regulate. And when we applied this distance criteria to the Gasparini data, we identified about 796,000 pairs of enhancers and corresponding target genes in the data. We refer to these as testable pairs of enhancers. And then we also looked at the distribution of cells in the Gasparini data that actually received guide RNAs targeting both enhancers in a testable pair. And what we found was that most such cases are only observed in fewer than 10 cells, which suggests that we may have limited power for detecting true interaction effects due to small sample sizes that we can provide to our model. And so to address this concern, I decided to perform a power analysis to try and answer two primary questions. The first question is, what magnitude of interaction effect sizes do we have the power to detect? And the second question is, how many cells need to actually receive perturbations to both enhancers in a testable pair in order for us to detect an interaction effect between them? One way this can be achieved in a CRISPR experiment is by increasing the MOI, because then every cell will just be receiving more different guide RNAs. And so I developed a data simulation framework to try to simulate ground truth data from a single cell CRISPR iScreen for enhancers. And this would allow us to evaluate model performance. And then I simulated data corresponding to different MOIs or lambda values, as well as different magnitudes of interaction effect sizes. And the resulting simulated data for the power analysis kind of has this hierarchical structure where I have a different set of ground truth data corresponding to each of the parameters that I tweaked for the power analysis. And what that analysis reveals is that we need a pretty high MOI experiment, which would lead to a large number of cells containing both perturbations that we're trying to model, in order to capture any small to mild interaction effects, although our false positive rate remains very low at all MOIs that we simulated. And what our power analysis also suggests is that for the Gasparini data that we're analyzing, which most closely resembles the lambda equals 15 MOI scenario, we may only have the power to detect larger interaction effect sizes. But nevertheless, we proceed with our analysis of the data with this caveat in mind, with the intention of seeing what we can discover in this data and to also demonstrate the utility of our model for future applications to data sets that are bigger and better powered to answer this specific question. Now, for our genome scale analysis of the experimental data, I'm going to be referring back to the testing criteria that I described on the earlier slide and those testable pairs that I identified in the Gasparini data. Based on our findings from their power analysis, we decided to restrict to a minimum of 20 cells in which perturbations to a testable enhancer pair was observed, leaving us with just under 4,000 pairs of enhancers throughout the genome, to which we then applied our interactions model to evaluate the presence of interaction effects. And among the cases that we tested, we identified 4 cases with significant evidence of an interaction effect. However, we suspected that these cases may have been driven by outlier cells or cells with extreme levels of the target gene's expression. And so to interrogate the robustness of our findings, we performed a bootstrapping analysis, sampling cells with replacement, to see if these results would hold up. And what we found was that after 100 bootstraps, these significant interaction effects did not remain significant, and we found that they were indeed driven by a cell that had a very high level of the target gene's expression. And so altogether, our findings from our work support a model where enhancers work together multiplicatively to control gene expression without support for any strong or common interaction effects between enhancer pairs. To strengthen this conclusion, we also fit an additive null model to a subset of enhancer pairs in the experimental data and found that in all cases, the multiplicative null model fit the data better. And so this histogram shows the distribution of delta AIC scores between the multiplicative and additive models. An AIC score essentially measures how well your model fits the data, with a smaller AIC score indicating a better model fit. So this distribution of entirely negative delta AIC scores shows that in all cases, the additive model yielded a larger AIC score and thus a worse model fit to the data than the multiplicative model. And our results also argue that any strong interaction effects between pairs of enhancers are not very common, and they do exist, and they're probably infrequently occurring, of small effect, or restricted to occurring in specific cell types or conditions that weren't observed in our analysis. And as for how our findings can be reconciled with previous reports of synergy or redundancy between enhancers, one possible explanation is that effects like those are naturally accounted for when using a multiplicative model of enhancer activity without requiring an additional interaction term, as would be necessary under an additive model of activity. And so now I'd like to wrap up my talk by summarizing some important outcomes of our work. First, we've presented a statistical framework for genome-wide interrogation of interactions between enhancers, from which future studies that examine enhancer interactions could benefit. And this is an advancement in the field because existing work studying this phenomenon has typically looked at specific gene loci rather than the entire genome. And we've also demonstrated that accounting for guide efficiency is important for accurately evaluating functional effects from CRISPR screens. And this is not the current norm when it comes to analyzing data from CRISPR screens, but our work demonstrates that it should be. And while we had limited power to detect any smaller interaction effects with the experimental data that we were working with, we do hope that our work can serve as a proof of concept for how one could perform this type of analyses to answer these questions with bigger and better powered data sets in the future. And in that vein, we hope that what we've managed to find here will motivate future CRISPR experiments that are specifically designed to interrogate enhancer interactions on a larger scale. And so now I'd like to thank my lab, especially Karthik, who is the undergrad that worked with me on the computational side of this project, and Zhou and Aaron, who are two former lab members who did those CRISPR-I knockout experiments for those enhancers in NMU that I talked about in the beginning of this talk. And I'd also like to thank my funding sources. And also there's a lot I had to leave out of this talk due to time, but our preprint is up on bioRxiv, so check it out if you're interested.
Well, thank you, Jessica.
Thanks, Jessica. Great talk. So can you elaborate a little bit more on the redundancy and how this model explained the redundancy?
Yeah, so in our case, we didn't see any sort of redundancy. Our interaction term is meant to account for anything that would deviate from like the multiplicative model. So redundancy, I guess, would, I guess if the effect were there, the interaction term should come up as significant. It might have like a different like directionality of effect, like positive versus negative. But that's like, so that we ruled it out because we didn't see any significant interaction. Yeah, we didn't see much of it here. But you know, it's possible. So like I guess I was saying, like maybe one reason that other people have reported that is because like our model, like we use a multiplicative model, and other people assume an additive model of activity. And so like you might not need additional interaction, like because the additive model works differently, you would require an additional interaction term to show that something deviates from like a linear pattern, versus with a multiplicative model, you wouldn't require that interaction term. And maybe that's why some people have concluded that. Also, I think some of these studies that have looked at specific gene loci, one of the benefits is they look at more enhancers. So also that's maybe something you see that arises when you look at more than two enhancers at once. Sorry for the long answer.
Here.
Hi.
Thanks for a great talk. Same question as the previous talk. Did you check the data set for off targets?
For what?
Off targets.
Yeah, so I guess our way of checking for off targets is to try to like measure the efficiency, but that's actually a really good question. It's related.
Off targets.
Yeah, it's exactly. It's that's actually one of the reviewer comments that we just got on this paper. So the next thing I will be working on when I get home from my vacation is trying. I think we actually need to account for both off targets and efficiency, because both of those together determine guide RNA performance. And I think that what we need to be accounting for in these models is not actually specifically guide RNA efficiency, but guide RNA performance, and it requires both of those components to do accurately.
Yeah, but if they're off target effect, you can't remove them from your analysis, right? So then you have to take out specific genes. So basically, the outcome effect you see if there is off target. How do you see then you remove it from your analysis?
I guess we didn't do that. We only evaluated efficiency. So yeah, I mean, we tried to do some experimental validations for like, you know, some of those enhancers, but I don't think we were really able to evaluate off targets there.
Hi, that was really interesting. I was curious, does your data contain cases where only one enhancer is perturbed? Because it seems to me like estimating the effect of the enhancer acting on its own, or the effect on when only one enhancer is perturbed, is like a good baseline for comparing to the joint perturbation. Like if you estimate the effect of a single when it is in fact a joint perturbation, is there something different going on there?
So, okay, I'm going to try and answer this question. Let me know if it's right. So you're asking if we looked at single enhancers like in the original Gasparini paper?
I guess I'm also not sure, but I'm just imagining that comparing to the effect of when the enhancer is perturbed and something else is also perturbed, you could compare to a situation where only one enhancer is perturbed.
So we did evaluate that. So it's kind of the same, like, so we try to evaluate something similar to what they evaluated in the Gasparini paper, which is looking at individual enhancers and seeing if we can like identify if they have any significant effects on any nearby genes. So we did that as well. I actually have some slides about it, but we mostly try to use that to try to evaluate how well our model is working, especially when we added this term to account for guide RNA efficiency. But I'm not quite sure if that's the same thing that you're asking.
Yeah, so I wonder if there's like a
the kind of baseline to detect whether you have an interaction is whether when this thing operates on its own, it changes its behavior as compared to when they're operating together. But
Or maybe you can clarify this in a coffee break. Okay?
Sorry.